All right, can you hear me? All right, that's good. Welcome, class of 1973, 50th year reunion. 50 years since graduation. It's incredible. Here we are once again. Welcome all the guests that are here and anyone else who's straggled in to hear John DiPersio talk about cancer. A rather unpleasant subject, but one that is with us, all of us, has been 50 years ago, now and into the future. And we look back and we look to now and we look in the future as we consider this subject. 50 years ago, this was a death sentence for so many, and many of us remember these things. And the treatment was to cut out the solid cancers and to poison it with harsh chemicals, and that was about it. We move forward to today because of people who worked so hard their whole lives on the research for cancer treatment and with the hopes of cure, and how we've gotten to the place where we are now. And that was through the hard work and dedications of lifetimes of people like Dr. John DiPersio, our classmate. So I'm going to tell you a little bit about John. He, right after our graduation, he went to the University of Rochester Medical School, and he didn't want to just take 4 years going through with just an MD. He stayed around for an extra 3 years to get an MD PhD in hematology and oncology. From there, he went on to his internship and his the one after that. Thank you, residency he sticks up for me. It's okay. At Parkland's Hospital in Dallas and the UCLA in Los Angeles. And there he was learning more and refining the techniques of high-dose chemotherapy with bone marrow transplantation support. And then he just took a job back in Rochester at the University of Rochester to run its nascent bone marrow transplant unit. And it was just by happenstance that I happened to be in Rochester. Now, neither of us are from Rochester, but there we were, and we were having a beer, and he told me he was having trouble getting insurance coverage for his patients for advanced breast cancer treatment. And we talked about it some. I did a little research, and we started to collaborate. And it turns out that John's many skills are not just researching and treating people, but getting on a witness stand and convincing a federal judge involved that in fact, the insurance companies had it wrong, and the treatment that was given was covered under the policies, which enabled a whole bunch more people to get treated. But I don't tell you that because of me or him. I tell you, but it gave me a chance to see what John is as a human being to the people around him in his workplace. It gave me a chance to really know his patients, to know his fellow doctors, and to know his fellow staff members. And I can tell you that the respect and the love that they had for him was palpable and inspiring. So, with that background, I go on to the future where he couldn't just stay where he was, but he got offered a job in St. Louis at that institution there, the Siteman Cancer Center at Washington University School of Medicine, and has been there ever since, running their program there and making great strides in both research and treatment, and globe-trotting around the world, giving speeches about what he does. So he really knows what he's doing. Fortunately, in his own life, he was able to when he finally got around to it, meet a wonderful woman, another cancer doc there, and marry her. And then had twins 12 years ago, Izzy and Jack. I know this because they were born 2 or 3 months before my first grandchild. So he was a little slow on that, but on the rest, it's been pretty amazing. So I won't waste any more of this precious time. John's to speak about that subject I can barely read. You have John DiPersio.
Okay. Now, I guess for those of you who know me, I think there is some implicit understanding of who I am. But for those of you who don't know me, remember I had twins 12 years ago. Think about that. It's like having twins when you're 90, you know, I mean, it's a Herculean effort, I'll tell you. So anything else I've done in my life it pales in comparison to that. You know, Bob, when I moved to Rochester, Bob did help me much, and he was actually, as you might imagine, he was the fiery entity that pushed these cases through the courts. He did help me get these patients approved therapies under the laws of the state of New York. But he also helped me get dates in New York, which was also another big part of his role. He's been a lifelong friend, and also I'm here with my two roommates from Williams, which is even more special for me. I mean, it's a wonderful thing giving this talk to all of you, but to see these two characters who made my life utterly miserable at Williams College is great. Now, my charge today is really to just give you a general idea about where the world of gene therapy and cellular therapy is going at the present time. And I just want this to be a quick review. I don't expect you to really assimilate everything I'm saying, but just understand some of the basic concepts. And I want to be around for a couple of questions in the end for any of you that may have them. Okay, now this thing is not going forward. Let's see. Let's try this now. Okay, these are my disclosures. So the first thing I want to talk about is the human genome. So I was a part of the Human Genome Sequencing Project. It was done by primarily 4 institutions: Baylor, the Sanger Institute in England, Washington University Genome Center, and the Broad at MIT. And when the sequence was finally annotated, this is what they found. They found that the coding region of the genome, for all of you who took biology at Williams, you know, you go from DNA to RNA to protein, and your genome represents all this DNA. And it turns out that only 1.3% of your genome actually codes for RNA and proteins. The rest of it is 3-dimensional structure, probably incredibly important. It's evolved over millions of years so that it supports the expression and regulated expression of thousands and thousands of genes. But the interesting thing is that the actual coding region of the genome is only a small fraction of the entire human genome. It's actually 26,000 genes, in fact, and about 2 billion base pairs. And so we sequenced that genome and then went on and said, we were at a bar one night in St. Louis, and the people that really did this were at the Genome Institute at WashU. And my colleague Tim Lay said, what about sequencing a cancer genome? And everybody said, oh, that's completely impossible. You know, and so we had a few more beers, and there were bets. How many mutations would there be in a cancer genome, because these cancer cells were so crazy? And so the bets ranged from 1,000 to almost 1 million mutations through this 2 billion base pairs of genome. And it turned out this was the first patient we sequenced. It was the wife of a physician at Washington University, and she had a standard risk acute myelogenous leukemia. She relapsed after a number of treatments and eventually passed away. And so when we sequenced the genome, we found only these 12 or 15 genes that were mutated, which was incredible. That meant to us that eventually we'd be able, this is a manageable number of genes that are mutated. We eventually, at least in this disease, will have some chance of really understanding some of the biology of the disease if we tear apart each one of those mutations and see how it affects the stem cell and how it actually contributes to acute leukemia. And one of the other interesting things is that this patient had, this patient had also several of these what's called single nucleotide polymorphisms. These are the things you're born with. And in the gene called TP53, which actually nobody knew about in this family, but actually puts her and all of her family members at much increased risk for malignancy. So she had these, they're called germline variants, and that contributed to the onset of this leukemia as well. So this was the beginning of what you've probably read a million times in the newspapers as the field of precision medicine. This was the first example of precision medicine in cancer biology by sequencing the human cancer genome. Subsequently, hundreds of thousands of cancer genomes have been sequenced. The entire sequence of the first human genome took about 11 years and was a joint public-private effort, and that effort cost about $2.2 billion. The first cancer genome sequenced at our place cost about $3 million, and now a human genome can be sequenced for $600. So we've made progress, and we've made efficiencies of scale, and this is due to really people that I don't even know how they think. These are called engineers, how they develop these machines to do automated sequencing and things like this, but they've made it possible for us to make these observations related to mutations and which mutations contribute to the disease. This is just an example of a patient who had acute leukemia, and you can see at diagnosis up here, you can see that these different colors are different combinations of mutations. We call them clones. So when a patient develops acute leukemia, it's not one clone, but it's several clones. And so this patient was treated with chemotherapy and went into a remission, but this one little clone persisted. We'd want to know, why did that clone persist? And how does it contribute to a relapse? So the patient was relapsed here, and when the patient relapsed, you can see that this major clone contributed to most of the first relapse here. But there was a new clone, a different color, that popped out of nowhere. This was actually in this at a very, very low level. And then when the patient was treated with a transplant from a donor, all of these things went away for the most part, and there were these new clones that emerged, 2 major new clones that emerged, but this gray clone persisted. So this is called the founder clone. So we understand how diseases progress, get initiated. There's a founder clone or a founder set of mutations that are in all the cancer cells. They contribute to it. And then they just start budding off little subclones. And those subclones can be the ones that become very aggressive, and those are the ones that kill you in the end. So what about curing this disease? Well, one thing that we've been focused on for a long time in many groups in the world, and you all live in major cities and you all have experts in your cities that do this, and it's called stem cell transplantation. This is a simple process by which the differences between the donor and the recipient can be used to the advantage of the patient. Those differences are what's called histocompatibility differences. And so the donor gets into the recipient and recognizes the recipient as non-self, and also recognizes the recipient's leukemia as non-self and attacks the leukemia. Unfortunately, some of these patients also have their normal tissues attacked by the donor immune system, and that can cause something called graft versus host disease, which can be mild, moderate, or life-threatening. But this is a powerful curative technique that we've worked on for many years, and now we're finally able to do this with much less toxicity. We're able to do much older patients. So I'm going to be transplanting a 76-year-old gentleman in about a week or two. This would be unheard of even 3 or 4 years ago. 10 years ago, we never transplanted anybody over 60 years of age. And you know, this is a very, AML and acute leukemias are more common in older patients. And so it actually allows us to access older patients who need it, and also we can do it in a safer fashion. So this is just a cartoon. I made this for you guys, by the way. I am not great on the computer, but you'll see how complicated this is. So look at that. So that's like an organ transplant. When you transplant an organ into someone's body, the immune system of the host attacks it, and that's what causes rejection if you get a kidney transplant. But the same process works for a bone marrow transplant, except the immune system is not in the host, it's in the donor. So it looks like this. So the transplanted graft goes in and attacks the host tissues. and that caused graft-versus-host disease, but that also can eliminate residual leukemia. It's very powerful. And we continue to hone this and improve this over time. So I want to talk to you about other approaches at immunotherapy. This is gene therapy, immunotherapy, and cellular therapies. One area that I thought I'd just tantalize you with is something called CRISPR, and you've been reading about this over and over and over again, and it is a very interesting technique. I'm going to give you some of the background, and you'll be kind of surprised. It's actually called clustered regularly interspaced palindromic repeats. I want you all to remember that. These repeats occur in about 40% of bacteria. So that's where they were discovered, in bacteria. And there's a gene in bacteria called Cas9, which is a gene that cuts up DNA. And then you can put these together, the Cas9, this enzyme, and so there were a group of investigators that actually noticed this in bacteria and fungi, and attached the enzyme to the sequence, and then designed RNAs, which could allow this whole complex to go right to a specific area of the genome. And the RNAs allow it to go to gene A or gene B, and you can direct it to any gene in any part of any gene in the entire genome. There's just one exception, which I'll mention at the end of the talk. In the whole world, all the genes in the human genome can be genetically edited except for one gene, and I'll mention that at the very end. So these guide RNAs actually hybridize and they match to certain sequences in the DNA, so you can make whatever RNA you want, attach it to this Cas enzyme, send it into a cell, it'll go to the exact spot you want, and it pairs up, and then this nuclease, this enzyme, cuts the DNA. And when DNA is cut, when both strands are cut, the repair process depends upon lots of things. And in your cells, that's why as you get older, there's a higher incidence of cancers, all sorts of cancers as you get older, because every time your cells divide, those repair processes have to be perfect. And if they're not perfect, then they'll instill mutations, which causes genes to be either overexpressed or deleted. And so what happens is that this process, when you cut both strands, is very imperfect. And so you get these things called indels, which are these areas where there's gaps in the genome, or you have extra bases added, which screw up the total sequence of a protein, or make the protein stop sooner than it's supposed to stop, and so there's no protein made. So, and they bind to this area called the PAM. This is called a protospacer adjacent motif with sequence. So these areas are just sort of landmarks, like a road signs for the guide RNAs. The guide RNAs have to see the sequence it's going for, and it also has to see one of these PAM sequences, otherwise the DNA cannot be cut specifically. Well, there's a whole bunch of ways to do this, and for many reasons, I'm just saying that CRISPR, which you've heard about, is the reason it's so prominent and prevalent today is that it's the simplest, cheapest, and easiest to use, and most dynamic, and so that's what's moved forward. But there are other ways to do this. And this is what the actual CRISPR complex looks like. There are a bunch of enzymes and components, and then there's this PAM interacting site which has to bind to a certain sequence, and then the whole thing sits down in the DNA based on where the RNA tells it to go, and then cuts the DNA in both strands of the DNA. And so this is what happens. So there's the double-stranded breaks, as I said, most of the time are repaired using this process, which is called non-homologous end joining, which is imperfect. Almost always the genes are messed up when this happens. And when they're repaired, they're repaired incorrectly and the gene is essentially deleted. And that's how we can go in specifically throughout the whole genome and delete any gene we want based on their sequence, because we can design any RNA that matches that sequence. Now, for homologous directed repair, that's different. We can send those into the right area, but in this case, we use a template, which is a big piece of DNA that gets replaced into it. So at both ends, we have these imperfect repair, but in the middle, we replace the bad gene with a good gene. That's called homologous repair. And that is much more inefficient, but it works. So this is sort of how it looks. You know, you can use a gene knockout where you get a double-stranded break, and then you get all this messed up DNA which deletes that gene. Or you can actually make cuts on both sides of a gene and knock it completely out. Or you can correct the gene by putting in a template, and therefore you put in the new gene for the bad gene. Or you can add a gene where a gene wasn't there, anywhere you want in the genome. So you can imagine with being able to do this, there's a lot of power and almost scary power of altering the genome of plants, animals, fungi, and even humans. Well, this is the timeline of how it was discovered, and I'll only say that the major observation was a long time ago, like many things. We think it's a new thing, but actually it was discovered in 1987. And the most important observations were made by these Danish food chemists, biologists, by the way. They were the ones that actually discovered what this whole process was all about. It's a bunch of crazy Danes in a food company in Denmark figured this out. I mean, who would have ever thought? They didn't get 1 cent from the NIH either. So, then two investigators, Emmanuel Carpentier and Jennifer Doudna, started developing ways of adapting this for human and eukaryotic cells. So not bacteria, but human and eukaryotic cells. And then Dr. Zhang at MIT was doing the same thing. Dr. Zhang and the MIT group filed their patent first. And for the last 10 years, there's been a monumental multi-billion-dollar patent suit between UC Berkeley and MIT over the patent rights of the CRISPR technology. And it was just recently finally adjudicated after having, I think, 4 separate appeals by the UC Berkeley group, and it was finally thrown out, and now the patent belongs to MIT. The one institution that doesn't need the money. So this is how it works. It's really amazing that we could adapt this for humans. It developed evolutionarily to protect bacteria against who is the worst? What's the worst thing a bacteria can see? A virus, exactly. They're called bacteriophages. They get in and they muck up the bacteria, they kill bacteria, they can kill entire strains of bacteria. So these bacteria have these sequences and they have Cas9 in them, and not all of the bacteria, but many bacteria. And when a virus infects a bacteria and injects its DNA in there, then it induces the expression of this RNA which binds to a sequence in the virus or to any plasmid DNA. And then they also make these Cas proteins, which are these same nucleases which cut DNA. And the RNA and the Cas proteins bind together and then go to the specific sites in the virus and cut up the virus DNA and kills the virus. Protects the bacteria against the virus. It's the immune system of a bacteria which is the foundation of modern CRISPR technology in humans today. So these are the guys that, and I told you the history, and Feng is a pretty happy guy these days, I would say. Okay, now I'm going to try to do this. Okay, I want to show you this little film which may say better than I could say it.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
We can go back to the regular slides now. Thank you. So just a little bit of comic relief. Another installment of Science Stand-Up. KFC has asked me to edit the chicken genome. They want something CRISPR. Never good at comedy, but so this is where we stand now. So that's the beginnings, the foundation, and the development of CRISPR to modify eukaryotic and genomes of living things and animals, including humans. And there has now been adaptations of this that have allowed this to really advance to an incredible way. You know, remember what I said, that CRISPR cuts DNA in both strands, and that really is a problem. Our cells are specifically designed not to accept that kind of hostile invasion. So we have all these mechanisms in our cells that when they see double-stranded DNA breaks, that they turn on a massive array of killer sequences which push the cell to die. So it's complicated. And also there can be rearrangements in the genome. You can get, with double-stranded breaks, you can get whole pieces of genome that are switched around and things. And so from CRISPR, gene editing has developed a process called base editing. Instead of cleaving the DNA, you just change the sequence of the DNA. It's pretty amazing. By putting in an enzyme, a Cas enzyme that is defective, but has a different enzymatic activity. And there are these base editors which change cytosines to guanines and et cetera. And they're called CBE and ABI editors, and they can just modify the genome without cleaving it. So it's much safer. So all of those rearrangements that might occur are minimized. So this is really exciting. This has just been developing in the last year or two. And over the past year, this is really dependent on just two kinds of enzymes, this cytosine deaminase and adenosine deaminase. But this will only affect 2 of the 4 base pairs. So this new approach called prime editing allows you to go in and change any base at any place. So now we have pretty much unfettered access to the entire genome of any species, and we can change any sequence to any sequence with high specificity without cleaving DNA. And so I don't want to show you this because it's just too much. This is the 3-dimensional structure of prime editing, which is an incredible advance in the field. And the way it's an advance is that these RNAs, instead of being very short, they're very long, and they also have, as part of the complex, a different enzyme called the reverse transcriptase, which allows these non-specific, these whatever you want to change the DNA to, occur at any base, all 4 bases. So you can do any change at any location as long as there is a so-called PAM site nearby. This is the same. These are the steps of prime editing, just to show you that it's complicated, and it would take me a while to go through this. And quite honestly, this is brand new. It's very exciting. Has its limitations, but overall, this will really change the game. So, what are the current and future uses of gene and base editing and prime editing? So you can see clinical therapies, improving the yields of genetic modifications, plant biosciences, disease modifications, drug development. We can generate all sorts of species that are inherently resistant or sensitive to various things to change the biosphere. And so one could argue, if you're a purist, that this is all crazy, and that any small modification of any genome in any species might have long-term detrimental effects. So I couldn't argue with you there. Obviously, the potential for good is great, and the potential for harm is there, and we need to be vigilant and look for it. So this is just an example of how gene editing has been used. Now, this is the cure of a disease called sickle cell anemia. So when we, normally there's no cure for sickle cell anemia, and transplantation can be done, which I talked about in the beginning, but that's associated with graft versus host disease. And older sickle cell anemia patients over the age of 20 have a very high mortality rate with transplant and also high complication rate. So you'd want to use it in the very youngest patients. But the very youngest patients, this will result in infertility and a lot of long-term complications. And so about 80% of parents of young sickle cell patients refuse to have their children undergo transplant because of those potential risks. But if we could do gene editing or gene therapy without all those complications, I think that would change the game. Believe me, many children with sickle cell anemia and many adults with sickle cell anemia are now being cured and treated with stem cell transplantation, but it's a tough road to hoe. And now we're starting to treat patients with sickle cell anemia using gene therapy. I'll just give you an example. So the way this works is that you, if I can get my pointer to come to here. Okay, so the first thing is that the cells come out of the patients with sickle cell anemia, and these stem cells get harvested and purified, and then they get gene modified. And then they get reintroduced into the same patient after the patient has received some kind of what's called conditioning or chemotherapy. And then we wait for those cells to engraft, and those cells will have had some genetic process done to reverse the defect. And this has now been effective and proven to be, at least over the short term, curative. These are the stem cells that we get to do this. They're very unique cells. They look like lymphocytes, but they can be purified from the bone marrow, from the peripheral blood of patients, and then they can be genetically manipulated. And so this is how you do it. There's different ways. So one way is to do transplantation, which I just talked to you about, and the other is to put a retrovirus in there with just an overexpression of a normal hemoglobin gene. It's called beta globin. That's being pursued by a company called Bluebird Bio. And Bluebird Bio has applied to the European regulatory agencies for approval, but not to the American regulatory agencies yet. It was approved in Europe for the treatment of sickle cell anemia, and the cost is $2.6 million per patient. The third approach is called DNA editing, and there's a gene called BCL11A, which what this does is it blocks the production of a hemoglobin called fetal hemoglobin, and more fetal hemoglobin actually reverses the effect of sickle cell anemia. And so it doesn't do anything to the gene defect, it just produces a different hemoglobin in the stem cells, which blocks the effects of the sickle gene mutation. And that's being pursued by a company called Vertex in collaboration with another company called CRISPR, cute name. And then another way to do this is to do the template approach, just take the completely unmanipulated gene and replace it with a bad gene. And that's a company called Graphite Bio. That turns out that study was initiated, and the study resulted in some significant genetic changes and a low efficiency of gene editing. And then there are several other ways of doing this. And then most recently, people are trying to do this in vivo, in fact, inject an adenovirus right into your blood without taking cells out. And those adenoviruses find their way to the stem cells, and they correct the defect in the stem cells. That is a decade or two away, if it'll ever work at all. So this is just how it goes. You just take the cells out, you manipulate them, and there are different ways to manipulate them, and then you assess their efficiency of correction, and then you inject it back into a patient, and you follow the patient over time. So this is a curative therapy in some patients, and right now this is going to be up for US FDA approval for at least two different techniques, the Bluebird and the CRISPR Vertex version in the next year, and the cost to the American insurance companies or to society will be somewhere between $2.5 and $3.3 million per patient. So, but we can also do other things. We can modify gene expression, so we can make animals that are genetically different so that we can study various diseases like multiple sclerosis or Duchenne's muscular dystrophy. We can actually use these CRISPR techniques to select for genes that modify certain phenotypes, so we can identify genes that we could intervene with to stop certain diseases, or we can actually modify the expression of various diseases using CRISPR. But there are problems, so that these, even though the guides tell where to go exactly, they can be off-target effects, means that they can go to other areas of the genome and cause some double-stranded breaks, which then might result in chromosomal rearrangements. And so there are a number of different problems that could occur. And the FDA is being very vigilant. It's incredibly expensive, about $1 billion to develop one of these techniques or more. And it takes several years, and so it can't be done in an academic lab. It has to be done with a corporate commercial partner. And so this is big business, big money, and also big risk. So these are some of the things that have moved forward in the world of clinical biology. So there have been successes with removing or genetically modifying mosquitoes so that they don't carry malaria. Sounds like a great thing. Yeah, maybe. Yeah, we've been able to eliminate cancer cells using gene editing, and I'll show you an example of something that we've been doing ourselves, which is very unique. Treating muscular dystrophy by injecting those adenoviruses in the body and allowing them to correct the dystrophin gene, which is mutated in enough cells so that in enough muscle cells so that at least animals have reversal of their symptoms, and this is going to be tested in humans in the next year or two. It's been used to alter the size and amount and milk production of cows in China. There are pigs that have been generated using multiple edits in which all of the recognizable pig genes on the surface of the pig cells are deleted, so that when we put a pig organ into a human being, there's no proteins on the surface of those cells which the human immune system can recognize. This work is being done by George Church at Harvard, and this has not been completed yet, but it's an amazing tour de force. HIV has been made resistant. In other words, you can do a stem cell transplant by deleting the gene for the receptor for HIV. So a patient has HIV and you can't get rid of it. You just take those stem cells, delete the gene for which the HIV binds to, and then those new stem cells are resistant to HIV, and they make T cells that are resistant to HIV, and so on and so forth. Mini pigs, if you want a pet in China, you can get a mini pig that's been made. They're little tiny pigs like this big, but they're cute as can be. And also important things like curing diseases like sickle cell anemia, and even editing human beings. So there's sadly one example of this. He Zhangqi, and you've probably read about this case. He did do this same HIV experiment in a human embryo and developed, and resulting in the birth of two children who had genetically edited genomes so that they could not be infected with HIV. And so obviously there's some theoretical positives there, but obviously he was lambasted and he was jailed in China for this. And the interesting thing was that he had, during his experiments, he had a number of U.S. investigators that were, giving him advice, and then when he finally went public with this, they all said, "Oh, this is the worst thing that could ever be." Okay, so I want to just say one example of how we've been working on this, and this has to do with CAR T-cell therapy and gene therapy. So, CAR Ts are these T cells. These are the immune cells in your body that attack tumors and fight infections. And so you can put genetic material in them so that they can be directed to specific tumor cells. And so this is what they look like. They have the outside of an antibody. Let's see here. The outside of an antibody here. And they have the inside, which is signaling the T cell to divide. And so when these things bind, and they're in a T cell to the tumor cell, they activate the T cell and kill the tumor cell. This has been a huge advance for the treatment of lymphoma and leukemia and multiple myeloma in the United States. And now thousands of these treatments are being done every month here in the United States. The question is whether society can afford to pay the $275,000 for each process. The product costs that much, and the actual treatment, because people need to be hospitalized, are another $150,000. So it's about $500,000 per treatment. And so this is the way it works. You get cells out of the peripheral blood, you isolate the T cells, and then you stimulate them, and then you CRISPR, or you put in genes in there like CAR Ts, and then you infuse them back into the patient and see what happens. Well, we were interested in a different kind of acute lymphoblastic leukemia, which is deadly, called T-cell ALL. And remember, the B-cell ALL, we use T cells to kill the B-cell ALL. But when you use T cells to kill the T-cell ALL, all the targets on the T-cell malignancy are also on the T-cell itself. So what would happen? What happens is that the T cells that are expressing that chimeric antigen receptor see themselves as the target, and they kill themselves. It's called fratricide, and they don't work. So no one's been able to do this. So we thought we could use CRISPR to figure this out. So this was done by a bright postdoc in the lab, and so the issue was that the target on the tumor cells is the same as on the T cells, and so this would result in fratricide. And also, the T cells in the peripheral blood and the malignant cells in the peripheral blood look the same. And so you're always going to have contaminating malignant cells when you do the genetic modification. And if you are successful in the genetic modification, then the tumor cells will be deleted of that target, and they'll be inherently resistant to the therapy. Or you could use T cells from somebody else. But Bob's T cells infused into my body, if I was given chemotherapy, would kill me like that because they'd attack me from this process called graft versus host disease. And so in order to get around that, this is just an example. This is a high-powered photograph of the peripheral blood of a patient with T cell ALL. And this is the target. We're going after these little diamonds. Can you tell which ones are malignant and which ones are normal T cells? This is a test. This is like an art history test, which is the fake. No, they're all the same. And so you can't tell the difference, but let me help you. So I just put an M on the malignant ones, okay? So let's say we do the editing and we, in this setting, this is what might happen, right? We edit out the target off the normal T cells, and now there's no fratricide because that cell doesn't have that target anymore, which is great. But still, what will happen is some of the malignant cells will be edited as well. And they will be in that mixture when it gets reinfused in the patient, and they'll be completely resistant to treatment because they don't have the target anymore. Now, if we use somebody else's T cells and we take the target out, then they're not contaminated with malignant cells, and we can effectively eliminate the disease that way. So that's what we thought we could try to do using CRISPR. And so to do this, we gene edited out this mechanism by which a T cell recognizes a foreign entity as foreign. This is called a T cell receptor, so we just deleted that by CRISPR. And we also took the target that we were going after in the T cell ALL off the T cell itself. And so there's no possibility of fratricide, and there's no possibility of graft versus host disease. And we can use Bob's T cells to treat everybody in the audience that has T cell ALL. And so this is what happens if you don't take the target out. These cells just sit there and die in the dish. And so we did a number of experiments, and we actually did CRISPR. We put these, we activated the T cells, and then we deleted both of those targets, the T cell receptor and the targets, which in this case it was CD7. And then we grew these cells up and then tested them. And here they are in the right panel, and you can see these mice actually have tumor in them that are T cell ALL, and they emit light, visible light out of the mouse's bodies as we put a gene in the tumor cells. So any cell, any mice that have this blue color, that means they have a ton of tumor and they're emitting a very high amount of blue light. And when we essentially treat those mice effectively, you can see that the light goes away. And so our CRISPRed CAR-Ts work beautifully, and this is what they look like. They express the CD3, which is a T cell receptor, and CD7. Normally, this is your T cells, and when we finish the editing process, which takes only a couple of hours, both the CD7 and the CD3 are deleted. So there's no CD7, there's no CD3. And we've done this with a number of different antigens. This is CD2 to treat other T cell malignancies, because not all T cell malignancies express the CD7. And you can see the cells for which we did delete and make these CARs. They're called universal CARs. They kill target cells that express that antigen beautifully. And these are irrelevant targets which aren't killed. Here are mice that develop a T cell disease, and we can cure these mice with these gene-edited CAR-Ts. And here's another example of how we can completely eliminate these tumors in mice using these edited. And so I'm going to finish up here with that was just an example of one way that we can use CRISPR to treat a disease which has been incurable and causes the deaths of lots of kids in adults in the United States and the world. And I think this is a way forward, and this will work. And it may not cure all of them, but it'll get enough of them cured and get enough of them to transplant so that many of them can be cured. And so I was just a few days ago I was invited to give a talk at the Fred Hutchinson Cancer Research Center. And these are all the luminaries of leukemia and cellular therapies. And we stood for a group picture at the end. No big deal. Notice that I have a full glass of wine here. That was my 4th glass of wine, I think. It was great. And there were 3 guys after the picture. We were just talking. And it turns out that Chris Horrigan, Ben Ebert, Chris, who was head of the cell therapy group at the NCI, National Cancer Institute, and he's a leukemia guy as well. And Ben Ebert, who is the chairman of the Department of Oncology at Dana-Farber, and myself are all Williams graduates, and we were all born on July 4th. You really can't make this stuff up. So this is really 3 generations of July 4th William grads. And I've stayed close. It was after CRISPR. This is a week ago. This is just to say that I valued my friendship with my Williams College over the past 50 years. I wouldn't be who I am if I didn't have those friends and colleagues. This is Tom Geisler and myself and 2 of my colleagues sitting in a restaurant in San Francisco. We were right around the corner from this guy. Recognize this? Yeah, it's the real Maltese Falcon from the movie. It's right there, was right beside us. And this is Tom, I think, sitting in front of Bob Sella's house here in Williamstown. This was on the 45th or 40th, I can't remember. Yeah, and because it's not so clear today, but look at that. We would walk around every day here and never pay attention to those mountains, and gosh.
You said that previous picture was only a week old.
Yeah.
Where's your whiskers?
No, this one. There they are.
No, not that one. Not that one.
Oh no, this is from before COVID.
Oh, all right.
Yeah. No, I grew these last night. So this is Tom, really. He's standing in front of in the Purple Valley. And this is us, which we'll do again this time. I'll beat him. I beat him just badly the last time. He can't stand being beaten. Marty, Doggett, Bob, and Geisler in front of Fort Hoosick House. That's where we lived. A great place. I went out there before last night. It's just beautiful. I wondered where all the rotten curved leaded windows were now. They're all like very neat, nice windows, and they all shut close to the. There. Here are the 3 of us. And Muggs, Richard. Yeah. Looked good. 5 years of chemotherapy. This guy is the strongest guy on the planet. God rest his soul. And this is in an airport somewhere, Bob. I don't know where. Koegel had to get in the picture. And this is us on the roof at Fort Hoosick just before graduation, the 3 of us. And this is, I don't know if there's any lacrosse coaches in the audience, but my son is addicted to lacrosse, and he's wearing a Williams hat, so in case, he's 12, but any early admissions would be great. And so I want to thank Williams College, and happy 50th to the entire class of 1973. And I feel like this guy. This is a friend of mine, Mike Eruzione. Does anybody know who he is? Forget about those Nobel Prize winners. This guy, no. Anyway, we were at a bar. It was late at night, and this is what he said to me. I said, I asked him, what do you, how has hockey impacted your life? And he said, it's changed everything. He said to me, I'm the luckiest guy in the world. If I had shot that puck 6 inches to the left, I'd be painting bridges now. It's true. It's true, because he would be, if he could even get up on a ladder to paint them. So I want to thank you. And one other thing, the other issue is that I told you that the entire human genome can be edited, with only one exception. And that's the one gene that can't be edited from our genome is the Williams experience. I'm happy to answer any questions as well, if you have any. Yes. Say your name, please.
My name is Alistair Mook.
Alistair, okay.
Hi. I'm also a father of twins, and you can come to my presentation on how to get them to 16 years old later. One of my kids was diagnosed with ALL B-cell when she was 5, and she's now a healthy 16-year-old. And we spend a lot of time talking to other parents to this day who have kids in treatment. And my sense over the 10 years, no one was talking about this gene therapy, at least it wasn't in our lexicon when we were in the hospital, but now I read about it all the time, but my sense is still that it's very much preserved for after relapse. It's a backstop. And maybe you answered the question, is that because of cost, or is it because traditional chemo is still the more effective?
For children, traditional chemo is very effective. Somewhere between 80 and 90% of children are with B-cell ALL. How old was your child when? Yeah, so that's the most curable age for ALL. So probably 90% of children with B-cell ALL are cured now. So obviously it's a risky procedure, and it's very costly. And it will slowly get moved more up front in the treatment, but it's way at the back, as you say, for those reasons. And as it gets moved up, we got to figure out how to make it cheaper. And so one of the things about this CRISPR technology is that I can make from you, if you have ALL and you had failed several therapies and you were going to get a CAR T cell, which is now approved by the FDA for adults and children. So if I were to take your cells and manipulate them, like I said, with just gene therapy for CAR T, and infuse them into you, the total cost would be $750,000, something like that. Very expensive. And so if I take a CRISPRed gene-edited allogeneic product from Robert, directed to that same target, I can make enough T cells from his peripheral blood to treat 20 to 50 patients. So 20 to 50 patients, the same process, everything's the same, except that one editing step, right, is added so that I can use his cells and I don't have to use your cells. So your stuff costs $750,000, and the question is, since there's been no allo products approved yet, what's an off-the-shelf allo product going to be? But they're going to be the same, probably $250,000. So there are ways to make this cheaper, and as they get cheaper, they'll be moved more in the front line. For CAR Ts for lymphoma, they were at the very end, and now they're being moved up and up and up, and now there are trials randomizing the upfront treatment for patients that don't get a great response after a couple of rounds of chemotherapy to continued chemotherapy versus CAR T cell therapy versus autologous transplant. So that's how it happens. Yes.
You gave an excellent, well understood talk.
It's the first time I gave it. I'm kidding.
Yeah, it isn't. But one of the calculations that I would be interested in is the cost of T-cell therapy relative to regaining productivity and economic advancement. Has anyone sat down and juggled those numbers?
Yeah, there are a number of studies that are ongoing looking at.
You're talking $200,000 or even $2 million, but if you
Yeah.
prolong the productive lifespan
Right.
of someone 20 years.
Exactly. So, yeah. So there are a number of studies that show that this is cost effective, even though they're expensive. And believe me, how these drugs are priced, they get a bunch of, who's an economist in the audience here? Any economists at all? You're an economist. Yeah, they got a bunch of guys like you that sit around this room and they don't let you talk to the press or the outside world at all. You work for a pharmaceutical company, and they really dive deep into every aspect of cost. They use Medicare databases, and so for lymphoma, they appear to be cost effective. Now, you might ask, how could a drug cost $2.5 million for sickle cell? But think about it. Sickle cell patients, they live an average of 50 years now, 52 years. That's a median survival. So normally in the population today, I think our average age is 73, 72.5, something like that. So, but sickle cell patients, their median age is 50. And so they live a long time. And during those 50 years, there are many different things that happen to them. Infections, hyposplenism, avascular necrosis, pulmonary hypertension, sickle crises, on and on and on and on and on. And they priced this, at $2.4 million because actually the health economists felt that the average cost of care for a sickle cell patient for their entire life was something like $5.7 million. So, obviously, they know what they're doing, and they try to price it that's reasonable. Unfortunately, what the public's response to all of this is, it doesn't matter if it costs $3 billion to develop, and it doesn't matter that the cost of standard care is $5.7 million. They just see that cost and they go, what? These guys are just crooks. But they're not really crooks. They're trying to maintain the viability of their businesses like everybody does so that they can invent new things. And you can't keep on going on if you are going into huge financial deficits. So, yeah, David.
The bill lasted 73.
Congratulations. You made it.
And also medical school with John at Rochester. So take us 10 years from now. Is this going to be the therapy of choice, or are these still going to be second, third, fourth, and
No, I think they're going to be the therapy of choice, and I also think that a lot of these genetic diseases like sickle cell anemia and thalassemia are going to be corrected in utero. Before even birth, for many reasons.
Well, we can talk about that at our 60th.
I told you I got off the plane in Albany, and I was suddenly full of remorse that my life was over, and it's the blink of an eye, and you're going your 50th reunion, and there are all these kids around you. You're almost dead. It's like the Monty Python movie, to bring out your dead, but you're almost dead. And so I said, this is, there's nothing after this. And some guy beside me said, is this your final destination? And I said, no. He said, well, where are you going? I said, well, I'm going to Williamstown for my 50th reunion. And there was suddenly the person behind me in the seat behind me goes, you're going to your 50th reunion? I'm going to my 55th. And then the guy right across the way says, I'm going to my 60th. So, yeah, yes.
Yeah, class of 68, Cornell Medical School. So we do a lot of great work with hematogenous tumors. Can you talk a little bit about solid tumors?
Oh yeah, I could say that this is a enormous challenge for immunotherapy. So I can tell you what my biases are. It's like trying to, if you compare your ability to destroy a water balloon and a rock. The water balloon, if you send something towards a water balloon and it has any kind of velocity, it can pop the balloon and the balloon will disappear. If you send an arrow or even a bullet towards a rock, what happens is that you chip off a piece of it and the rock's still there. But on the other hand, if you surround that rock with some kind of abrasive and you keep on doing this every 5 minutes for 3 years, that rock will whittle away and disappear. So this is the difference in how I think the efficacy of checkpoint inhibitor therapy, which is, for all of you that don't know about this is an antibody-based therapy that basically tells the immune system to be unblocked. And so the immune system is activated in the context of these things called neoantigens. And they stick there. And so the T cells can get constantly activated. They constantly kill. They may die too. Some of them become memory T cells and they come back and they come back. And it takes a long time, but they whittle those tumors down. And for the CAR T stuff, the problem is that they are fast risers and fast decliners. There's a lot of activation-induced cell death, meaning that they go quick, they do a lot of damage if they can, and then they die. And in solid tumors, you need sort of more of a constant approach to the disease. And the tumors are, there's geographical and topographical and immunological barriers. There's a lot of other things that really have precluded these immunotherapies working for solid tumors. And people are still hopeful that we'll be able to use CAR Ts or natural killer cell CARs or other immune therapies to treat solid tumors. I'm a little less optimistic, and I think that these ways of enhancing the innate immune response to these minor antigens or neoantigens might actually work better. I don't know if that answers the question or not.
So the key thing, I guess, is going after the residual disease, so that this doesn't eliminate the need for effective primary therapy.
Yeah. So that's the other thing that, remember the fishbowl plot I showed you with all the colors? So residual disease, I hate to say this, but we all have residual disease. We've all generated multiple cancers. 80% of men above the age of 80 will have evidence of prostate cancer, and the same for breast cancer. But I mean, I think that we're generating cancers all the time. The longer we live, the more we generate. And so these cancers have to be eliminated by our immune system. And a lot of them can stay quiet for a long period of time. I just had a leukemic patient who was 20 years post-transplant and then relapsed. 20 years. So that's those cells, and it was the same clone, same colors as was present early on. Yeah, so minimal residual disease is, we'd like to get rid of it, but I think what we have to maybe think about is that we have to actually deal with it and how to sort of arm our immune system so that we can live with minimal residual disease so it doesn't, get a jump on us. Yes? Yeah, go ahead, try me.
Well, I'm not going to do the morality of for-profit medicine because people should make a living.
Yeah.
All right, but there's got to be some equity in this somewhere.
Yeah.
But,
I agree with you. I've been working at a university my whole life, so I agree with you.
Right. Federal government, we obviously know they're involved in R&D in the medical industry, and I'm wondering, do they ever think about putting a pool of money aside, billions of dollars, for the for people to get the treatment? Not just for you guys to figure it all out, but then to help pay for until the price can come down.
They do have various mechanisms. Obviously the budgetary process is very rigid, and so HMS has a certain amount of money that they can spend, and they have to, get money from different sources if it looks like they're going to go over those budgets. And so it's really hard for the government to do that without an act of Congress.
The other question I had is regarding medical marijuana. If you guys want to read my bio, you'll understand why I'm interested in marijuana still. I smoke my way out of Williams. On a serious note, the federal government still, marijuana is still illegal, all right? And I honestly believe that that is probably one of the greatest medicinal plants on the planet. Is there any research into medical marijuana with respect to cancer treatment?
Oh yeah, there's a gazillion things going on. I'm not involved in any of it, but yeah, there's a gazillion studies going on with medical marijuana. Medical marijuana is largely, approved in most states. And there are many physicians that I have a license to write prescriptions for medical marijuana in 3 or 4 different states. And I do it from time to time because some patients just don't get benefit from conventional medications. And they say that they are improved. So I believe that there's probably some patients that benefit from it. But as far as sort of rigorous studies showing a true anti-cancer effect of medical marijuana, I haven't seen any.
That you're talking about applies to what you're talking about.
Yeah.
Does any of that happen in?
No, I don't think so. Somebody has to fund that, and that may be.
A lot of institutions would have done whatever, but they can't.
Yeah, well, that's true because you need money to do research, and so your research ideas have to be considered competitive by your peers, and if there's not enough peers that think that that's a competitive project, they're not going to fund you. All right, I think that's it. Thank you very much.